resources. So that's what we are doing at the Quantitative Biology Center in Tübingen, QBIC, which is a bioinformatics core facility running all sorts of analyses, genomics, transcriptomics, metabolomics, all sorts of data from individual research groups and research institutes. And what we're trying to do is we want to help research scientists to analyze their data efficiently, ideally in a fair and reproducible way. So that's why we actually started picking up Nextflow for doing that, and also always try to leverage resources such as, for example, de.NBI resources to do the analysis, because we don't have enough hardware on local premise, on premise basically, to do these kind of analysis all on our own, although we have our own HPC as well. So as most of you already know, data in computational biology, but also physics, chemistry can be really big. So typically it's very big data in terms of also sometimes, physics, this is kind of the master example for that. There you have sometimes petabyte scale data. In biology, we're not yet there. Maybe if we accumulate that, we might actually hit that as well. In larger two-digit petabyte scale, we typically have to deal with diverse data. So we have sequencing, proteomics data in the biology field, for example, which is what we're mostly doing at QBIC, for example. And what we typically also see, unfortunately, is that we have erroneous data, meaning, for example, it contains sequencing errors. We have problems sometimes with the instruments that we try to use to measure certain biological experiments. So we have to deal with that as well. And given that these are likely to occur if you have really large data sets, because they occur with a certain probability, you can be very certain that if you have large-scale data, that you see these errors popping up at some point, and you have to be aware of that. And that all leads me basically to that kind of central paradigm here. We need methods and tools to analyze such big data in an efficient way, in a scalable way, ideally in a way that also biologists will initially and also finally understand what you're actually doing with that in a bioinformatics setting. So one challenge that was briefly also mentioned in other talks is that we have the problem with software dependencies. So that's completely from a bioinformatics perspective given here. What we typically have is we have workflows or pipelines that try to do certain data analysis consisting of various different tools. We have dozens of individual methods that have been built by somebody else, that have been actually compiled in various programming languages. And we have these binaries and we want to do some analysis with that. So what we're ending up in these kind of pipelines then typically is a complex dependency tree and also very complex configuration requirements for these pipelines. So that is actually a nice little plot showing a companion web server application that uses several dozens of tools to do an annotation of a parasite genome and the analysis of that parasite genome. So if you just have a look at this huge mess, actually, it's actually not a mess. They actually did a pretty good job in actually making this web service available, but it's a mess if you have a look at this in the first place. And that's what you're typically seeing in the many bioinformatics applications, and bioinformaticians around can probably kind of say, I've seen similar things in my life already. So, and that's what you have to live with. Another problem with these software dependencies was actually found nicely in a paper which was now published in June. A lot of the tools that are actually out there in bioinformatics seemed difficult to install, and 28% of the tools even failed to be installed completely. So that was kind of a random selection of tools out there by a group, Mangul et al. It's published in PLOS Biology. So what they did basically, they took a couple of hundred of tools that are kind of commonly used in bioinformatics, and they tried to install these and get them actually up and running, which is kind of the everyday work of a bioinformatician. If it were not for projects like the BioConda and Conda Forge projects, for example, that help you in doing these kind of difficult things. But still, there are tools out there that you can't even get into Conda or BioConda because you simply can't compile them, for example, for various reasons. Sometimes it's really not possible anymore because there's some missing C++ header files that are just on a hard drive of an old PhD student that has been gone for 10 years or something like that. Stuff like this happens, unfortunately. So that's what we have to live with. Another problem that we see now more than in bigger projects as well is that large-scale projects are coming more frequently in bioinformatics. So it started with projects like the 1000 Genomes project, which is more or less finished, let's say. There are projects in the UK like the 100,000 Genomes projects. There is even a draft call for the EU 1 million Genomes project, and various other projects are actually popping up all over the globe. So what people are actually facing in these with these big projects is that they have to be able to reproduce what they've been doing a couple of years ago. Because if you want to truly integrate analyses that you have been doing like 2 years, 3 years, 5 years ago, with these large data sets, you can't produce that at a single time point. You can't do that from today to tomorrow. You have to do that over years, accumulate the data, and being able to do the same thing that you did with the very first genome you created in that project with the last one as well. So that's what you want to do. So you need reproducibility to do these kind of projects. And it's really crucial to be able to do that. So Björn was actually mentioning a couple of issues with that, how you can achieve that and how you can do that. And actually, I'm just stressing this point more and more, because this is what people have to live with. And this is just human genetics in this case, but we also see these kind of projects in all various other types of applications in genomics, but also metabolomics and transcriptomics, for example, and other fields as well. So what you have to be able, you have to make it clear that all of your papers that you publish should also be able to be reproduced at any given time point in the future. That's the ideal case that you would like to see, hopefully at some point. And what we see today is that a lot of the papers published in journals, for example, are really hard to reproduce, if at all. So what we see is, this is actually a nice illustration from GigaScience, which was put on Twitter a couple of months ago. And what people typically see in biology, we see a nice fancy method out there that some fancy bioinformatician, theoretical biologist put up there. This is cool. I want to try that method on my fancy new data that I just created with a nanopore sequencer, for example. And what they want to do is basically run that. And then you just start digging and you find out, okay, this is just running on a specific operating system. This is just running in a specific version on the data that I just have, because they used a different protocol, for example, to create that. Then you have problems with the language. You can't translate it, for example, because your compiler doesn't work with a specific version, for example, because you have a C++ library or something like that in place. So there's lots of caveats, which this illustration is just kind of trying to grasp. That might not be entirely complete, of course, but it's just kind of a fun way to show, okay, there's more than just trying a method. It's typically down to what you actually have to do in the overall process to get this running. So with that in mind, I'd like to introduce a bit Nextflow, which we at Cubic now try to use since a couple of years to do all sorts of analysis, big data analysis. Nextflow is a domain-specific language that you can basically learn quite quickly from scratch. So it's a Groovy-based language, which allows for fast prototyping of workflow of a pipeline. You can basically do task composition, so you can even have individual tasks that do certain things in that language, and you can easily parallelize what you're doing. So if you have certain things that can be parallelized easily, for example, you have like something that an individual process that just takes a FASTQ file, does something with it, and you have multiple FASTQ files, this is automatically parallelized if you have the computational resources for that. So if you have 50 of these and you have 50 cores on your HPC, on your de.NBI cloud, for example, then this can run simultaneously on 50 cores. It doesn't care about that, actually. That's quite nice, because you don't, and that's actually something you don't have to do on your own. It's automatically done by the language. It's self-contained, so it can use containerized tasks. So each of these tasks can be put into a container. So you can have all of your dependencies in a container, for example, using Docker or Singularity. You can also use other technologies like Rocket nowadays. And it also is nicely playing with isolation of dependencies. You can keep these containers, rerun the entire analysis, just keeping the Nextflow script and your Docker container. You can rerun the entire thing as the same way on all kinds of infrastructures that you want to use, which brings me back to the usage of de.NBI Cloud, for example. So you can basically develop on your local laptop, you can run that on your local workstation, take the same thing and put it on de.NBI and run that there easily. That's quite neat, actually. So to sum that up, it's completely automated. You can automate an entire pipeline that does certain things that you want to do. It parallelizes certain tasks if this is possible. It's quite reliable, so we're running hundreds of projects at Cubic alone, and we have lots of other collaborators in different locations, for example in Sweden, people in Barcelona, some other labs at the Francis Crick Institute are also running a lot of Nextflow tasks. It runs quite reliable. It's one of the more reliable ones, workflow management systems we've been seeing around. And it's very easy to learn. So we have a lot of biologists, project managers who are not bioinformaticians, who are not actively developing these pipelines that we're using, but are actually actively using them. So they just take their FASTQ, whatever data they have, for example, from RNA-seq, and they run them using the standard pipeline that we provide. And our hope in the end is also to automate this process in the future at Cubic, at least. For standard projects, this is going to be completely automated, so that whenever a sequence is done with RNA-seq data and we have a certain design for that, this is automatically running through a pipeline. Nice fancy fact again, which I already mentioned, is everything is completely reproducible because the container is versionized on a Docker hub, and we have a versionized version of each workflow, so we can basically go back and say, okay, I want to run version 1.0 of the RNA-seq workflow at some point, and that will automatically fetch the accompanying container required with all the dependencies that we use. So we kind of solving that problem. Nextflow uses a data flow model, which in the beginning, if you develop Nextflow scripts, is a bit hard to grab. But at some point, after a couple of hours of investigation, you typically get it quite quickly. For me, it was kind of a natural way of thinking about things. So you have individual processes, such as BWA here, for example. Your data flows in, basically the BWA process takes it, does something with it. The data flows out of the BWA process and flows to the next process, in this case SAM tools, and you can combine that arbitrarily. So you can have that kind of stream of data, and you can divide that in substreams to different modules that do something with your data. So that's kind of the concept behind this data flow model. And that's very easy and a logical, natural way to understand how these scripts basically work. So what you have, for example, you have your input here. So on the left side, you see a small Nextflow script, which is a very easy one. Basically creates a channel from data, from file pairs. So it takes a read input data, so FASTQ files, for example. It takes these as input. It creates an output, a FASTQC, zip, or HTML report, basically, and pushes this to a next channel. And the script just does FASTQC minus Q on these read files. And that would ideally, given that FASTQC is installed, of course, would simply run this completely and analyze your data. So that's a very, very small example, actually. And the code to run that actually can be seen on the right side, on the top. So if you go there, Nextflow run main and F, you define the read star FASTQ or something like that. That's all you need to do, basically. And then you run the entire thing. You can do that with Docker Singularity or with Bioconda support. So if you have your dependencies like FastQC, for example, is available on Bioconda, you just define minus with Bioconda or user configuration profile, and this is automatically fetching the required Conda packages, for example. Or you do the same thing with Docker Singularity. The nice thing about Nextflow is you can do a so-called process executor abstraction. So you write your script, you write it in a way that you just would write any bash script, for example, on your local machine. And what you do is you can run it locally by defining the executor to be local, but you can do the same thing by defining the executor to be PBS toric, for example. So if you have no local machine to test this, you can also configure a cluster scheduler. That's just 3 lines in a configuration profile. You don't actually have to change your main script. So you just have a profile, you have your main script, and that's it. And the cool thing about this is you can run the same code on various types of infrastructure without actually meddling around with the main source code. You just define profiles for different types of infrastructure. That's it. You separate the actual workflow code from the infrastructure side, which is really nice because you can port the same code to various infrastructures. So we've been using the same RNA-seq workflow without actually changing the workflow code on local infrastructure workstations that we have, collaborator workstations, cloud resources such as de.NBI Cloud, but also other HPC infrastructures, cluster systems that we are having access to, which is really nice because it improves the code portability. You don't have to reinvent the wheel every time you change your infrastructure. So with that in mind, we figured out, okay, this is nice. It's nice to have Nextflow because it really helps a lot in developing these kind of things, like pipelines for certain types of analysis. And then we figured out with other people who are doing the same kind of work in bioinformatics core units, okay, let's put this one level higher. Maybe it makes sense to create a community out of this because we're all kind of doing the same types of analyses like RNA-seq analysis, genomics, whole exome, whole genome sequencing analysis. And what we wanted to do is basically, okay, let's just put all of our wheels together. Let's not develop 3 individual RNA-seq workflows, one in Sweden, one in Tübingen, one in Freiburg, one in Ulm, wherever you want to go, basically. Let's just build one big one that can basically accommodate all the needs of all these different bioinformatics core units and make that better. That was the basic assumption of this. So the cool thing about this, if you all contribute to one larger objective, it's much easier to save time in developments, invest that, save time in more testing, more frequent updates. New method comes out, some people are taking care of testing the method on actual real data, figuring out, okay, this is maybe better than what we've been using it, making it available to the larger community in a workflow, for example, so that others can also contribute and improve their own work. So that's what basically led to NF Core. NF Core is a community of people basically who are starting, similar to other open source communities, to collect workflows for all sorts of analysis tasks in computational biology. We also have people from chemistry now picking this up, proteomics and metabolomics, but the main focus was so far genomics and transcriptomics. We have 20 plus institutions who were already joining and contributing actively, either by just documentation if they're just users. So we have people who are simply just users who are using these pipelines. But we also have a lot of people who are developing their own workflows, contributing workflows from their own core units where they are actually working, and trying to improve already existing workflows to help the wider community. NF Core in numbers, I'm not going to go too deep into this. We have a pretty nice web page nowadays. So we have a nice pipeline overview. So if you want to do certain types of analysis, you can actually search that pipeline web page. This is interactive. So if you fancy doing something like quantification of peptides from mass spectrometry raw data, for example, you can use the MHC-Quant pipeline. We have some 16S rRNA amplicon sequencing analysis workflows, for example, with lots of information on the individual workflows. So if you go there, you basically see, okay, this is how you could run that workflow. You see how many people were cloning that workflow in the last 2 months. There's lots of statistics who contributed to that workflow. There's an introduction, some documentation on the actual workflow. You can basically get quite a quick grasp on what is already provided at NF Core, and if you don't find anything that helps you with your specific analysis task, you can just go there, join the Slack community, for example, ask for help, ask for somebody who might have started working on something that helps you, for example, which happened quite often nowadays. There's also developer and user documentation. So if you want to contribute a new one, we have guidelines. The possibilities to add a new pipeline is also there, of course. And there's lots of information on usage and other helper tools that we have there. NF Core itself wouldn't be as easy to do without the help of other upstream or downstream projects. So for example, without Bioconda and CondaForge, thank you guys, whoever is here, it wouldn't be that easy for us to package, for example, dependencies. I mean, this is like making that entire process much easier for everybody. Initially, we had our own like Docker files. We tried to do that on our own from scratch, basically, like everybody else did at a certain point, I guess. But then we figured out this is much easier using CondaForge and Bioconda. So that's what we're doing here. We're actually actively also encouraging everybody who's building a pipeline in NF Core has to follow certain types of guidelines. And one of these guidelines, for example, encompasses that you have to put all your dependencies for your workflow into CondaForge or Bioconda. That's a requirement in the NF Core community nowadays. So that means it automatically also contributes to other communities. In this way, we try to communicate that. We have a pretty open communication channel. So we have a Twitter account, we have a Google group, we have the NF Core webpage with lots of documentation, GitHub organization, and a big Slack channel that is actively used by I think we're like 170 users now at the moment on the Slack channel, users and developers. So that depends on the day how many people are actually online. So there's lots of people helping others as well nowadays. It's not just a core team of main contributors, but it's a bigger community nowadays, similar to other projects like Bioconda or CondaForge. As I already briefly mentioned, NF-Core enforces certain guidelines, which we found to be kind of a very important part of the project, because they enforce certain types of behavior, enforce certain types of requirements that help you actually building pipelines and workflows that last longer than just a couple of weeks. So one thing is that we basically now agreed NF Core, Nextflow Core, so it's all Nextflow based. We require all of the pipelines to be MIT licensed. Software has to be bundled in Docker Singularity / Bioconda / CondaForge. We require continuous integration testing of all the workflows. So each of these workflows has a Travis CI linked automated test with test data attached. So your workflow will be automatically tested whenever you change something on the code before you do a release, for example. What we also require is stable release tags. So pipelines have to have releases at a certain point. So if you do, let's say, a new workflow that does some fancy RNA analysis and you actually finish that, basically you can put that up for review. Lots of people will review this. And in the end, after you changed it to a way that it fits all of these guidelines, you do a release with a certain release tag. And then you can refer to that workflow 1.0.0 at any given time point in the future. And that's the idea behind that, for example. We require common pipeline usage and structure and software to be bundled in Bioconda. Cool thing with the tests is that NF Core has certain tests in place for all of the pipelines. So what we do here is we first of all lint the pipeline code, so that checks whether the guidelines are actually met, license is in place, Nextflow version is actually existing, stuff like this. So you can't actually make things wrong or get feedback. Okay, this is not the way it should be. We also lint the documentation. Linting documentation means that the markdown code that you write for your documentation is actually valid markdown. So your documentation can be parsed and rendered correctly in a web browser, for example. That is automatically tested. And then we require at least one test case for each of the pipelines that I mentioned. So that's quite easy. In this case, you just see it here, Nextflow run, Travis builder, and then profile test, Docker. This is automatically fetching test data from a test repository on GitHub that we have, a central one, fetches that data to a Travis instance and runs the pipeline using this test data to verify whether the pipeline actually runs through, at least with a given small test data set. We also have bigger test cases using Jenkins nowadays in place. For some of the bigger pipelines that are more mature, let's say, we have big test data sets available that are running a couple of hours, which we can't do on Travis for certain reasons, because the limit, I think, is 120 minutes or something like that nowadays. And therefore, we can't do that on Travis. So the entire infrastructure looks like that, the NF Core project infrastructure. We bundle software using Conda or Bioconda, package that into a Docker container for storing it as well, for permanent storage, archival storage in this case. We use GitHub for code management, so Nextflow can pull code directly from GitHub, or any GitLab service, for example, is also supported, Bitbucket as well. We test it using Travis CI, and then you can execute whatever you're doing in your pipeline on all kinds of different HPC, local infrastructure, de.NBI cloud, whatever you have actually. To make it easier for people to start using and building pipelines in NF Core, we have so-called NF Core framework tools, which is kind of inspired by what people were doing in the Bioconda team, but also other communities. We have tools that create a skeleton code for creating new pipelines, for example. So if you want to build a new pipeline from scratch and you're a newbie, you typically are like, okay, where do I start? This is, let's say, typically a quite difficult experience at the beginning. And what we have there is NF Core Create. So it's a small pipeline tool that you can install using Conda or pip or something like that. You install that locally, and then you just type NF Core Create, type enter, and it will be asking you a couple of questions like, what is your workflow about? What do you want to do? And then it creates a skeleton code, which is perfectly valid documentation, links, markdown documents, test cases. Basically, it's kind of a skeleton to build your own pipeline on top of it. and you just fill out the to-dos basically, and that's it. And then you have your workflow. So it makes it much easier to onboard newbies to the NF Core pipeline project. We have NF Core Lint, which I already briefly introduced, which just checks your code. So that helps you when you develop your pipeline. You can run NF Core Lint on the pipeline, and it will basically detect certain common errors, like, okay, you forgot to indent lines in your code, for example, in a weird way. For example, you get some feedback on that. At the moment, we have 15 stable pipelines, 19 in development, which is currently growing a lot because we have a lot of different institutions now jumping on the train, basically, starting to contribute their own pipelines or improving already existing ones. So back when we jumped on that train, for example, 2 years ago and started initiating together with Phil Ewells from Stockholm, we had like 5 pipelines or something like that. And for example, to just mention it briefly, the RNA-seq workflow in the beginning, for example, just had 2 options. That's it. So there were 2 methods that you could basically choose from. Now we have like 4. A 5th one is now being implemented. So somebody is taking care of that at the moment, which is great because you just come back from vacation like I did a couple of days ago, and you just find like 20 open pull requests to pipelines that you're actually using in your daily work, which is nice because you don't have to do it on your own. You just have to review now and help people making it stable. But that's nice. You're not alone anymore. Which I briefly, something I also mentioned briefly, which makes it much easier to run these kind of pipelines on various infrastructures, is the configs repository that we have. So remember I briefly mentioned that you can separate your infrastructure from the actual workflow code. So if you have your workflow, like let's say defining what you're doing with your data, you don't want to mess around with this. For example, remember what you did like 5 years ago with Bash. If you have to run a certain Bash script on a different infrastructure on your different workstation, you might have to fiddle with paths, binaries, locations, and stuff like this, which you don't have to do anymore with Nextflow. But still, you need to define how your infrastructure looks like at a certain point. So for example, if you have a torque scheduler, you need to define, okay, that torque schedule has a queue, this queue has certain maximum parameters and stuff like that. So that's what you have to do. And what we do is we share these configs in a central GitHub repository, and all NF Core pipelines can look up these shared configs. That means I contribute a single configuration for the Baden-Württemberg Cloud BNAC cluster, Bioinformatics and Astrophysics, and all NF Core pipelines can now use that configuration automatically. That means the RNA-seq workflow can now submit jobs to this cluster. The MHC-Quant workflow can do the same. All pipelines that are built using NF Core framework can now submit jobs to that cluster because this central location just defines how this cluster looks like, basically. Now this is just defined here. You just have basically some config description on top. Then you define that you want to enable singularity. You define that you load a certain singularities version on that server, on that cluster. You define the executor to use, like in this case, a PBS torque. You define the queue that you want to use by default. And where you store reference data, for example, how much memory you have on a typical node there, that's it. Same applies if you use de.NBI cloud instances, for example, you just have the central location, you host the config profiles there, done. All the NF Core pipelines can use that, automatically fetch it from that central location and be run on that kind of infrastructure, which is quite neat. You don't have to do that in each of the pipelines nowadays. So if you want to run Nextflow or NF Core workflows and de.NBI infrastructure, what we found is that you actually have 3 ways to do that. The first one is that you use a predefined, pre-compiled image and just spin up your instance that you want to use to run Nextflow NF Core. So for example, we had a couple of assembly projects which require a lot of memory. For these, I just needed a single node that runs these kind of jobs all the time. For these, I just created that image. I just booted one of these images, set up Nextflow quickly using this existing image, which is basically already containing Conda, Docker, and Nextflow, so it's very easy to work with. And then some of our project managers just connected there and run the job, basically. That's quite easy. If you want to have a bit more fanciness, then there's another option. You can also run a private Kubernetes cluster and spin that up using, for example, KubeSpray on top of the OpenStack that de.NBI provides. And the other option would also be set up your own cluster using, for example, bbgrid, that was already mentioned in another talk today. So these are basically your options how to run this on de.NBI resources if you want to. All NF core pipelines come with interactive reports, so that's what we typically also require here. So that's a bit more fanciness. These are interactive, so you can also create these with various types of other analyses. This is just the RNA-seq one, but you can also know the whole exome one, but you can also have these in various types of other workflows as well. Documentation, I already showed that in the interactive version, and we have a lot more that you can explore on the web page actually. What's next with NF4? So there are a couple of things that we're currently not very good at. One thing is that we currently use our own kind of way to build Docker images for workflows and pipelines. What we want to do is integrate here with the biocontainer community more efficiently. So our idea was to use biocontainers in the future for all the dependencies, since these are automatically built already for each of the bioconda recipes. We want to use that. We also have some ideas to do automated cloud tests, for example, for AWS price estimates. That's one thing that we were thinking about. And we want to have full-size testing for all pipelines. So that means that we want to run not just a single small test cases that we typically run in all the NF core pipelines, but also like full-blown realistic data sets for all the pipelines in an automated fashion using Jenkins, which we're currently looking to do that with de.NBI resources, because that's kind of the resources we'd need for certain things like that. And some of the ideas also center around modules. So Nextflow at the moment is a very monolithic thing, so you can't import certain blocks, building blocks. So at the moment you write your script, that's your script, then you can run it. But the Nextflow team is currently working very actively on modularizing that. So you can write a FastQC module, put that in a public repository, and then just import it from there, like a library function, basically. And that's the idea, to use that very, very soon. SnakeMake, for example, can do that since quite some time. And the idea is that we want to build that kind of community resource for people in an NF core modules repository. And yeah, of course, we want people to jump on the train, either by using pipelines or developing or putting documentation. We have a couple of people who are actually not developers, not really active users, but are having fun, for example, making figures and graphs for interpreting results, because that's what they do typically in their day. And I have a couple of people, for example, at the Max Planck Institute in Jena, who are now just drawing figures by hand to just display, okay, this is how an experiment would look like if it fails. This is an experiment that just went very well. And are putting these actively as contributions to a pipeline that I'm building, which is also nice contributions. So quite happy if you have that in such communities. Yes, so if you want to join, contribute, use it, or have some questions, just follow up on this. Tomorrow is going to be a Nextflow NF core session. So if you want to start using that, or might want to have a more in detail look, please come by, step by, and we'll have a deeper look at how you can build your own workflows, for example. Thank you.